Hello. Thank you for joining EssenBioScience today for this webinar. My name is Dan Appledorn, and I am the R&D Project Manager at EssenBioScience, and am the main contact for application development and support. I just want to mention a few items before we get started here. In the bottom left-hand corner of the screen, you'll notice a box that says Chat. You can certainly type in questions along the way or at the end of the presentation. You can type in questions, and we'll spend about 10 minutes of time answering those questions that we can get to while on the air. And if we don't get to your question, we'll certainly follow up with you in a personal email format and answer those questions. Second item is we will be playing a few videos throughout the course of the webinar, and you can change the quality of those videos by using a drop-down selection tool in the bottom right-hand corner while the videos are playing. And I'll remind you of that once we start playing some of these videos. This is the first webinar in a series of webinars that EssenBioScience will be presenting on a monthly basis. So please check back at our website for future webinars and future notices. Our subject for next month's webinar on Thursday, May 30th, will be Essen's Discovery Services, and that will include discussions about both ion channel services as well as live cell services using the IncuSite live content imaging platform. The title of today's webinar is Cell Player Kinetic Assays, focusing on proliferation, and also a quick mention of other assays that we currently offer, including migration invasion, and as well as cytotoxicity and apoptosis assays. Before launching into a detailed discussion of how we use live content imaging to measure proliferation in a kinetic way, I want to first take a step back and talk, at least conceptually, about the idea of live content imaging and provide you more of a definition. Live content imaging, as defined by EssenBioScience, is the acquisition, analysis, and quantification of images taken from living cells, cells that have not been fixed and stained, but rather cells that remain unperturbed by the staining or labeling or detection method. Because these cells remain unperturbed, this allows for repeated measures over longer periods of time within a physiologically relevant environment, such as your incubator. These periods of time can last from days to weeks. We believe that live content imaging is composed of a number of different attractive attributes from several different technologies or biological approaches. Those include live cell imaging, of course, live cell imaging being the spatial or temporal real-time resolution of cellular phenotypic changes as they occur over time. Live cell imaging allows you to record a history in either movies or image libraries that allow you to go back and review those images over time and identify and validate your quantitative data, as well as to discover different phenotypic changes and anomalies that you would otherwise miss if you're doing simple endpoint assays. This can lead to new discoveries that you would not otherwise have seen. Live cell imaging also allows you to take repeated measurements within the same well, which can do two things. Number one, increase the signal-to-noise of your assay, making it more precise. And secondly, can eliminate initial time course experiments that often take up a lot of time and resources. And of course, with a live cell imaging approach, the best place to keep your cells during these experiments is in your standard tissue culture incubator, where the environment is stable and physiologically relevant. Live content imaging also incorporates the most attractive features of high content imaging. This includes multiplexing assays, allowing you to derive more complex biological models, automated image processors that process images on the fly, leading to quantitative data, quantitative kinetic data. And this kinetic data can also inform plate-based pharmacology analyses. And these analyses can use methods that you probably haven't used before to determine pharmacology using kinetic data, such as area under the curve, peak responses over the time course of the assay or a concentration response, or informed single time point measurements, where you pick the time that's most relevant for processing your data or analyzing your data. But beyond the complexity of live cell imaging and high content imaging, we also bring in very simplistic approach to light microscopy, using light microscopy methods. For example, providing high quality phase contrast images in a software package that's designed for researchers at all levels. In this way, we make subjective standard cell culture measurements, like confluence measurements, into a more objective science, so that you can make decisions on when to split or not to split your cells simply by looking at the confluence of your cells and not making a subjective call without a scientific aspect. Of course, to make this possible, you need to support several different vessels and manufacturing vessel types, plates, flasks, as well as slides and different tissue culture dishes. This software, like plate reader software, is very simple to use, simple software interface, and the analysis tools are very easy to use and allow you to process images on the fly and graph data in real time so that you can make up-to-the-minute decisions on how you want your assay to proceed or not to proceed. We believe that we can apply this live content imaging approach to very simplistic biological questions as well as highly complex biological models. And it is really the combination of all of these different technologies and biological methodologies that make up the concept of live content imaging. And the glue that really holds all of this together is the simplicity by which the EssenBioScience IncuSite Zoom provides to users interested in taking advantage of live content imaging. The IncuSite Zoom system is a fully automated two-color phase contrast and fluorescence microscope. As pictured in the right-hand side on the top, this microscope is designed to work inside a standard tissue culture incubator. It supports several different vessel types, including flasks and dishes and plates of all different sizes and shapes. And for this reason is ideal for this long-term live content imaging approach where you collect kinetic, quantitative data over time. Jumping into a discussion on how we use live content imaging and Essen's reagents for kinetic proliferation assays. The image you see behind the title here are HT1080 cells that have been individually infected with a lentivirus expressing nuclear-restricted red fluorescent protein or nuclear-restricted green fluorescent protein. And we'll talk in more detail about these reagents and how we've used them to quantify proliferation of cells over time. This first video includes MDA MB231 breast cancer cells that have been labeled with the Nuclite Red reagent. Again, you can control the resolution of these videos using the drop-down in the right side bottom corner of the screen. And as I play this video, you'll notice that there's a very homogeneous labeling of the MDA MB231 cells, each nucleus containing a nuclear-restricted red fluorescent protein label. This label is stably integrated into the MDA MB231 genome, so the label is passed through doublings or passed through generations in order to facilitate the labeling and the quantification of the proliferation of these cells over long periods of time. This labeling strategy becomes obviously becomes the main aspect in counting the number of nuclei over time as a measure of cell proliferation. So how do we apply these reagents and labeling methodologies? This first slide here is a discussion on real-time cell counting in two colors. So in this case, we've labeled two different cell types, HT1080 fibrosarcoma cells, as well as A549 lung epithelial cells with either Nuclite Red reagent for HT1080s or Nuclite Green reagent for A549 cells. Enumerating these cells over time allows us to calculate doubling times of multiple different cell types in mixed cultures or co-cultures. You'll see in the image on the left that there's a nice homogeneous population of either green or red labeled cells. And then our automated image processing software allows us to segment those images based on their fluorescence values. So you can see that the blue and the pink segmentation masks have shown up over the fluorescent labels in these two different populations of cells. We then use those labels to quantify the number of cells over time as described in the graph at right, where the y-axis is the nuclear count, the left y-axis, I should say, is the nuclear count, and the x-axis is the time in hours. And as you can see, clearly HT1080 cells double at a faster rate than A549 cells, with a doubling time of about 16 to 18 hours for HT1080s and 21 to 22 hours for A549s. In addition to processing the fluorescence channel, the IncuCyte Zoom also has a trainable phase contrast processor that is able to segment the phase contrast image as well. So here you see a yellow segmentation mask overlay on the image at left, describing where we've detected cells within this image. This metric is exported at right as an HD phase confluence metric, and as you can see following the black line in the graph, that confluence increases in value over time until it reaches a saturation density of around 95 to 100% confluence. Interestingly, the nuclear labels, even though cells are at 100% confluence, the nuclear labels indicate that both HT1080 and A549 cells continue to proliferate even though the culture has reached saturation. Another interesting aspect of using nuclear labels as opposed to using confluence measures of proliferation. The next video I'll show is this same co-culture, HT1080 and A549 cells that have been labeled with nuclite green and nuclite red reagents. These are the original images that were used to quantify the number of nuclei at every given time point. And as you can see, as the experiment progresses, so does the cell count. And then these values can then be used to calculate rates of growth, doubling times. And eventually, we can use the area under the curve measurements or maximum value measurements to incorporate a pharmacological approach to measuring the effect of different compounds on inhibition of cell growth. So speaking more to that, this is an example where we have taken real-time cell counts and translated that into informed pharmacology. So I'll start from on the left side of the graph and move to the right. The first graph I'm showing on the left is indicating cell number over time. The y-axis is nuclear count, the x-axis is time. And here we've taken HT1080 fibrosarcoma cells that have the nuclite red reagent and have treated them with increasing concentrations of the cytostatic compound cyclohexamide from 4 nanomolar up to the highest concentration, which is 1 micromolar, and compared that to the vehicle control. If you focus on the open circles in that graph at the left, you'll see that cells continue to proliferate at a very rapid rate with a doubling time of about 16 to 20 hours if you actually do the math. And increasing concentrations of cyclohexamide significantly affect the proliferation of these cells, with the highest concentration almost completely inhibiting proliferation. If we translate the data on the y-axis to a log phase scale, we can see nice linearity in these readouts, justifying the use of an exponential growth fit to calculate the rates as shown in the middle graph, with a rate constant of the vehicle control, a k-value of 0.043, and the highest concentration of cyclohexamide, illustrating a k-value of about 0.01. Those k-values can then be plotted as they are at the far right graph on the y-axis as a function of the log of the cyclohexamide concentration expressed in molarity. And that these values can then be used to calculate pharmacological measures such as IC50 values or PIC50 values, whatever is most interesting to you. And not only do you get the quantitative readout, or the quantitative aspect of the data, which is obviously for all of us extremely important, but another feature of the IncuCyte strategy, or the live content imaging strategy, is you get qualitative information as well, or a historical record of what your cells look like following treatment with different compounds. For example, in the video at the left-hand side, these cells have been treated with vehicle control compared to cells on the right-hand side that have been treated with the highest concentration of cyclohexamide, which is 1 micromolar cyclohexamide. You'll notice that the cells on the left-hand side proliferate very readily. Obviously, they've not been treated with anything, and so continue to proliferate. Whereas the cells on the right-hand side have been treated with 1 micromolar cyclohexamide, and you can see a dramatic difference in their proliferation rate just looking at the videos as a qualitative assessment. Another interesting aspect of this is you can get a nice readout of cell morphology, showing that cells on the right, although they do not proliferate at the rate of the vehicle control, that their morphology is not too different from vehicle control treated cells, indicating that this is a drug that inhibits their growth but that does not induce a significant amount of cell death. Of course, it would be easier if you had a marker of cell death with the ability to multiplex this assay. And of course, we do provide that strategy, and we'll talk more about that in the slides upcoming. So how do we do this? Our cell player kinetic proliferation assays utilize these nuclite reagents, which I've preliminarily described. These reagents come in 2 flavors, the red fluorescent protein that is nuclear restricted or the GFP, green fluorescent protein that is nuclear restricted. We provide these in a lentiviral delivery system, thereby giving you high transduction efficiency and low toxicity. It is a VSVG pseudotype lentivirus, so it does get into a significant number of cell types, and they've been optimized in order to do so. Images are taken in the IncuSight Zoom live content imaging system, where you can capture time-lapse images and record these videos. We take images in both phase and fluorescence to look at both the confluence values as well as the nuclear localization of the red or green fluorescent protein. And this, of course, is all occurring within your incubator in order to provide the most physiologically relevant and stable environment. The live cell imaging approach, we can accommodate both immortalized cells, either transiently or stably using these viruses, as well as primary cultures. And then the software tools that are provided within the IncuSight Zoom system allow for automated image processing as well as real-time cell counting. As a package, this enables kinetic cell proliferations assays on up to 6 384 well plates per IncuSight Zoom. So why use live content imaging and nuclite reagents? I think most of us can agree that there's really no good, robust, information-rich kinetic method to quantify proliferation. Current methods that are used include indirect endpoint measurements based on either viability and/or metabolic readouts. These can include MTT assays or ATP assays as surrogates for cell metabolism or cell proliferation. If you do really want to achieve real-time cell counts, you can do that. Oftentimes, however, that's labor-intensive. It requires you to plate a significant number of plates or dishes and then pick each one of those up at individual times and run them through a cytometer in order to get an actual count. Other ways to measure proliferation, even within the IncuSight Zoom, include confluence measurements. Other systems use impedance-based measurements, and these work well for the most part, but can oftentimes show nonlinearity, especially when treating with growth factors or drug compounds that can result in a morphological change in the cell, where cells will flatten or grow and will be read out as an increase in cell size or read out as an increase in proliferation when it was really just an increase in cell size. And so these nuclite reagents and getting a nuclear count avoids that problem and gives you a real-time cell count or real-time nuclear count as opposed to confluence or impedance measurements. You can also imagine using existing chemical stains such as cell tracker stains, CFSE, or other DNA stains or protein-based labels. However, these stains are oftentimes suboptimal for many different reasons. The most important reason is a lot of these stains can affect the biology of the cell, either affecting its proliferation, their ability to differentiate, or some other biological effect that is off target and one that is not of interest to you. Other dyes that don't have a biological effect can often be not appropriate for use in the live cell imaging system. For example, you'll get nice segmentation of your cells at the beginning of your growth curve, but by the end of the growth curve, it's either diluted out or it has not followed along the lineages of the cells, and so you can't segment those images at the end of the assay. The nuclite reagents have been designed to address these issues. The reagents are extensively tested and validated at EssenBioScience, incorporating a number of different assays as controls. We've looked at the morphology of cells, comparing the parental cells to the daughter populations that have been infected with the lentivirus product. We've looked at their proliferation compared to parental controls as well as their migration. And in our angiogenesis assay, we've measured the ability for our HUVEC cells labeled with nuclite green and nuclite red to differentiate into complex tube networks. And the angiogenesis assay will be the subject of future webinars. Other cell types that we've looked at include HT1080, HeLa, MDA, MB231, A549, as I mentioned, the HUVEC cells, and our customers continue to use these reagents on a variety of different cell types, and as of now have not ran into a cell type that we've seen significant physiological abrogation when using these reagents. We then applied these reagents to a more basic research question. The question we asked was to determine the role of stromal cells in the resistance of breast cancer cells to lapatinib. This was a study that piggybacks off of a study in 2012 that appeared in Nature by Straussman et al. In this publication, they showed that breast adenocarcinomas that overexpress HER2, that the drug lapatinib can inhibit HER2 and EGFR pathways. They showed this particularly is true in SKBR3 breast adenocarcinoma cells, who by their very nature overexpress HER2 and are inhibited by lapatinib in a monoculture assay. And you can see an image of that was taken on an incucyte system in the top right image, where SKBR3 cells in monoculture are treated with 550 nanomolar of lapatinib. Suffice it to say that the morphology of these cells is dramatically different than untreated cells. However, when in co-culture with stromal cells, either CCD1068SK skin fibroblasts or human mammary fibroblast, HMFs, that these cells rescued the SKBR3 cells from lapatinib-induced inhibition when compared to the same cells in monoculture. And this is shown in the image at the bottom where SKBR3 cells are shown in co-culture with the 1068SK skin fibroblasts. And you can see the morphology of these cells and their organization within the culture is very different compared to those cells in the monoculture. Using these images, we then quantified the pharmacology of lapatinib. But before I move on to that slide, I want to discuss first how we generated the SKBR3 nuclite red cells. We made these cells by using the nuclite reagent, which incorporates a single EF1 alpha promoter that is used to express both the fluorescent label and a puromycin resistance marker with an IRES sequence in between, separating the two open reading frames. Day 1, we plated the cells in a flask or a plate. Day 2, we added the nuclite lentivirus reagent. Day 3, we simply replaced the media. And days 5 through 12, we applied drug selection, in this case puromycin, to derive a stable homogeneous population. Once this population was derived, we used these cells in the assay that I will discuss further. We set this experiment up all in one 96-well plate with an N of 4 per condition. The first 4 columns of the 96-well plate are SKBR3 cells simply plated in monoculture. The second 4 columns are SKBR3 cells plated in co-culture with human mammary fibroblasts. And in the last 4 columns, the SKBR3 cells are plated in co-culture with the CCD1068SK skin fibroblasts. Lapatinib treatment was applied from top to bottom, increasing concentrations at the top, highest concentrations at the top and lowest concentrations at the bottom, with the bottom row being the control-treated wells. Focusing first in the graph at the left, this graph shows the comparison of SKBR3 cells that are treated with a moderate level of lapatinib at 500 nanomolar, the y-axis being the nuclear count, the x-axis being time. And as you can see, SKBR3 cells without the presence of stromal cells or SKBR3 cells in monoculture do not proliferate. They are shown here as the purple line, whereas cells in combination or in co-culture with the skin fibroblasts and the mammary fibroblasts continue to proliferate. Although it appears that the skin fibroblasts provide a little bit more protection from lapatinib treatment than the human mammary fibroblasts, as suggested in the Straussman publication. In the figure at right, The y-axis represents the area under the curve of these kinetic graphs using the nuclear count metric. The x-axis represents the log of the lapatinib concentration, and as you can very easily see, SKBR3 cells without stromal cells, which is the purple line, have almost a 2 log higher sensitivity to lapatinib treatment compared to the SKBR3 cells cultured in the presence of either human mammary fibroblasts or in the 1068 skin fibroblasts, suggesting that the tumor microenvironment or the context in which the SKBR3 cells are growing has a dramatic effect on the sensitivity of these breast cancer cells to lapatinib treatment. This obviously has a lot of implications on the use of these compounds in vivo. The proliferation assay can also be miniaturized into, I showed you a 96 well plate example. This is a 384 well plate example. In this example, we've used HT1080 cells that have been labeled with the nuclite green reagent. And in this example, we are looking at 16 different compounds from top to bottom in the 384 well plate. And these are done in replicates. So a concentration response curve in columns 1 through 11 are the first replicate, and then the same exact treatments are applied to cells in columns 12 through 21 and 12 through 22, and in columns 23 and 24 are the control columns. With the IncuCyte system, again, we quantify this data kinetically over time, and you can graph this data at any given time point using our microplate graph function, or you can put each individual graph on their own XY axis. It depends on how you want to visualize your data. Here we get a really good idea of the whole plate and how the assay progressed with each compound simply by looking at the microplate graph view. And you get a pretty good idea of the quality of the data that comes off the system and the repeatability of the information just by comparing the left side of the plate to the right side of the plate. They're almost mirror images of each other. This data, again, can be used to determine pharmacological responses of the compounds. Again, on the y-axis in this figure, we're measuring now maximum object count, or peak response, if you will. On the x-axis is the log of the compound concentration, and for each individual compound, then we can calculate an IC50 curve. We can then compare those IC50 values between each compound and use these values to rank order these compounds in their ability to inhibit cell proliferation. But not only do you get a quantitative readout of the biology, but you also can use the images as a historical record to look at the difference in the morphology of the cells that are treated with these different compounds. For example, as you can see, doxorubicin does not seem to affect morphology of the cells, whereas treatments with staurosporine or 10-DEBC causes complete abrogation of the cell morphology and is likely inducing cell death, in addition to the possible effect of inhibition of cell growth. And differentiating between those two is always a challenge, and I'll talk more about that when we multiplex assays. So summarizing the cell proliferation data that I've showed you, our cell proliferation assay uses nuclite reagents which have been validated and extensively quality controlled. They provide high transduction efficiency and low toxicity and can be used in several different cell types. The data is quantitative and kinetic in nature, and movies and image libraries can be made to validate your observations in order to track morphology and phenotypic changes. Of course, the incorporation of multiple cell types in co-culture experiments and labeling each of those cell types facilitates the creation of complex biological models where you can start to ask additional questions that you weren't able to ask before. These assays can be miniaturized into 384 and 96 well plates, which allows you to interrogate directed compound libraries in up to 6 384 well plates at one time. And our software tools allow for automated data acquisition and integrated metric calculations, which can provide a convenient access to complex data. In addition to the single color examples that I showed you now, I now want to move towards differentiating the inhibition of cell growth from induction of cell death using two different reagents, one available through Life Technologies, the reagent Yo-Yo-1, and another reagent available through EssenBioScience, which is a measure of the activation of caspase-3 and caspase-7. And these reagents are used in order to differentiate between the cytostatic effect of a compound and the effect that is cytotoxic. First, the cell player two-color cytotoxicity assay incorporates the nuclear marker as a measure of cell proliferation, and this can be multiplexed with the Life Technologies reagent Yo-Yo-1. Yo-Yo-1 is a cell-impermeable DNA dye and gains access to the cell once the membrane becomes compromised. Once it gains access to the cell, it labels the DNA green, and then using the IncuCyte quantitative tools to identify green objects, we can count the number of Yo-Yo-1 positive objects and differentiate cells that are living and cells that are dead. In the video I'm showing here, these are HT1080 cells that are labeled with the nuclite red reagent. They are cultured in the presence of the cytotoxicity reagent Yo-Yo-1 and treated with 100 nanomolar camptothecin. And two things catch my eye in this assay. Number one is as camptothecin is added, that the cell motility is significantly decreased, so they stop moving. And then shortly after their inhibition of movement, you begin to see rapid cell death, as indicated by the green fluorescence marker. This can be quantified over time, as shown in the graph at the right, where we're quantifying Yo-Yo-1 object count per millimeter squared as a function of time on the x-axis. This data can be plotted, like I said, in several different ways. Again, here we're showing the microplate graph view. On the left-hand side, these are HT1080 nuclite red cells. The y-axis is the cell count. The x-axis in each well is time. And as you can see on the left-hand side of the plate, columns 1 through 6 are treated with the cytotoxic compound staurosporine, and columns 7 through 12 are treated with the cytostatic compound cyclohexamide. And using the nuclear reagent, we can see that the cells do not proliferate rapidly in rows A, B, and begin to proliferate a little bit in rows C for cyclohexamide, but are still significantly inhibited by staurosporine. If we then turn to the green channel, which is a marker of cell death, you can see that it's not only an inhibition of growth, but staurosporine induces a dramatic cell death response, whereas cyclohexamide, even at the highest concentration, does not induce a significant amount of cell death. There's a little bit there, but not as significant as treatment with staurosporine, indicating that cyclohexamide is an inhibitor of cell growth, but not an inducer of cell death or a cytostatic compound, as opposed to staurosporine, which is clearly cytotoxic. Our statistical analysis of this assay has revealed that we get a Z-prime factor of over 0.6, so it's sufficient for screening protocols if that's of interest to you. And we can do this in as many as 6 384 well plates at a time, giving you 2,300 wells of kinetic data per 24-hour period. We use a similar technique for measuring apoptosis. However, in our apoptosis assay, the green label is a DEVD-based reagent that when caspase-3/7 gets activated, it cleaves the DEVD motif, resulting in DNA staining by a DNA dye. And so once caspases are activated, then the cell turns green. In a similar way, then, we use that green marker as an indicator of caspase activity or apoptosis, and using the nuclear marker as a measure of proliferation. In this experiment, we've taken HeLa cells, labeled them with nuclite red, and treated them again with staurosporine. If you focus first in the top left corner, that is a microplate graph view of caspase-3/7 count. The top of the plate being the most concentrated staurosporine treatment, the bottom of the plate being vehicle controls. And as you can see, high concentrations of staurosporine induce a significant amount of apoptosis. If you look at the bottom, focus on the bottom left microplate graph, that is an indicator of the cell count over time, and so you see an inverse correlation to the caspase-3/7 count, indicating that staurosporine not only induces significant amount of cell death, but inhibits cell growth. These data can be plotted in an XY fashion. Analyses can include area under the curve or peak responses. In this case, we're using the area under the curve analysis to plot the pharmacology as shown in the graph at the right. The red indicating the nuclear count over time, the green indicating the caspase object count over time, and we can use that data then to calculate EC50 values for staurosporine and its ability to induce apoptosis, and IC50 values for the ability to inhibit cell growth. Again, the Z prime factor in this assay is greater than 0.7, allowing 638 well plates or 2,300 wells of data to be accessed within each 24-hour period. Now, a little bit outside this area of nuclear labels and proliferation, I briefly want to measure another assay that we perform at EssenBioScience. In this case, it's a label-free assay. It is a kinetic measure of both cell migration and invasion. This assay incorporates the 96-well wound maker and Essen image lock plates, so a consumable that is sold by EssenBioScience. And this is a label-free method for measuring the migration of cells in a modified scratch wound methodology. We use our live content imaging system to measure the movement of cells within the scratch wound area. That data can then be plotted, and area under the curve analyses or other methods for pharmacology can be used to calculate IC50 values for inhibition of either migration or invasion, all within the same 96-well plate. As an example of that, this shows the pharmacological effect of GM6001 on the invasive phenotype of HT1080 cells as they migrate across a thin layer or invade through a 3D layer of collagen 1. The y-axis in each one of these graphs is the relative wound density. The x-axis is time. And as you can see, in an invasion approach, GM6001, the broad MMP inhibitor, can prevent or inhibit the movement of HT1080 cells into the wound region in a concentration-dependent manner, whereas there is no effect on cell migration. These assays, the migration assay, cytotoxicity, and apoptosis assay, will be discussed in more detail in future webinars. And so at this point, I'd like to conclude just by summarizing live content imaging within the IncuCyte Zoom system. We believe that with live content imaging approach using IncuCyte Zoom can provide more sophisticated in vitro models, that you can gain increased mechanistic information, and that these co-culture or mixed culture experiments can begin to interrogate different signaling pathways, including crosstalk between two different cell types in a paracrine signaling type fashion. We believe that you can get full temporal resolution of biological events from viable, happy and healthy cells. And that can be supported in every well that contains a time-lapse movie that you can go back to and look at and validate your quantitative data. This data is analyzed on the fly, and so you can get access to it up to the minute or up to the hour. And these repeated measurements increase the signal-to-noise and precision of your assays and eliminates the necessity for initial time course experiments, which can be a burden on time and resources. These data can be used for quantitative plate-based pharmacology, and of course the software interface is designed in a way that is accessible to all users at all research levels so that you can analyze your data from your desk in a simplistic way. You can access this technology either by purchasing an instrument, an IncuCyte Zoom itself, but if you're not ready to do that, or if you want a validation of our system and the data that we get, we would encourage you also to take advantage of our discovery services. We provide discovery services for both live content assays, so live cell imaging experiments, as well as ion channel assays. And our 20 biologists that we have on staff with over 100 years of drug discovery experience, we are in a unique position to provide our expertise to answering your most challenging biological questions. Again, future webinars will focus on other aspects of live content imaging, including those that I've mentioned already, as well as incorporating angiogenesis assays, and our most recent release, which is a NeuroTrack assay to measure neurite dynamics, both retraction and extension of neurites. You can, of course, get more info on the technical specs of the instrument by downloading our IncuSite Zoom brochure. Of course, that is also available on our website, as well as all of the requirements, the hardware requirements. And I'd like to also say that available on our website are very detailed application notes that describe how we do these assays, including the assays that I was unfortunately not able to talk about in depth today. All of that is available at our website by downloading our application notes, and I encourage you to use that resource. If you have additional questions, feel free to email us at sales@essenbio.com. You can call us at 734-769-1600. Again, my name is Dan Appledorn. I'm the application scientist here. I'm the application scientist here and the manager of some R&D projects. Thank you very much for your time today, and I hope you found this informative. And stay tuned for our future webinars. We'll be doing this on a monthly basis.